So let's turn to the DNA structure and try to understand what it is that didn't escape Watson and Crick's notice. So first we will take a closer look at the building blocks of the DNA. So you probably already know. We have a phosphate group, we have a sugar group, and we have 4 different bases only. So that's the reason why people thought that DNA is too simple to explain the richness of heredity. So the 4 bases that we kept talking about all the time are depicted on the slide. Actually, when we go through the DNA, we will also take along the components of the RNA polymer as well. So they seem to differ minimally. You may have already spotted the difference between the thymine and uracil. So the uracil is lacking this methyl group. It seems to be a small change, but these small changes at the end actually make a big difference, as we will see later. Same is true also for the sugars used in DNA and RNA. At the very first sight, they all look similar. But when you look closely, then you will notice that the 2' carbon atom in DNA has an hydrogen atom, and the RNA has an OH group instead. So the 2-deoxyribose, so the name contains the syllable oxy. This may suggest that some oxidation is going on. It is actually exactly the opposite. From RNA to DNA is a reduction. And this is in fact also the physiologically relevant sequence of events. The cell makes the ribose first and reduces it to the deoxyribose. Why this is 2-deoxyribose, we will see later. And we are almost done. So we need to couple the base with the sugar, in this case the adenine. This happens over the 1' carbon of the sugar. That gives us the adenosine. The only part that is now missing is the triphosphate. This also couples to the sugar via the 5' carbon. And we have put together our first nucleotide, adenosine triphosphate, or ATP. You probably already heard about ATP before, or the letter A. But I want to really make sure that what we have put together here is not yet the DNA building block. So here we have the ribose sugar. This needs to be reduced to the hydrogen, and that is then our building block for DNA. If we then do this for the remaining 3 bases, we then have our genomic alphabet ready to go. A, T, C, and G. Remember, A stands for ATP. So if you wanted to polymerize DNA yourself, you wouldn't go to the Sigma catalog or whatever and buy ATP. Instead, if you want to have the building blocks, you buy the deoxynucleotides. All right. Next question is, how do these letters now line up? To understand that, we first look at a simple schematics. So we see here the building blocks of the DNA. We have a sugar phosphate that couples to a base, and that gives you the nucleotide. In this particular case, guanosine triphosphate, or G. And these letters then line up according to the Watson-Crick base pairing, C to G, A to T. So far, I am assuming I didn't tell you anything new. This depiction is now considerably more complex than the previous one. So we now see many details. First and foremost, of course, the Watson-Crick base pairing between the G and the C and between the T and the A. So either 3 hydrogen bonds or 2 hydrogen bonds. That specifies basically the base pairing. But we also see many other details. For instance, we see that this phosphate sugar backbone is labeled as a phosphodiester bond. We also see cryptic numbers 5' and 3' and next we will try to understand what all these specifications now mean. Okay, let's take a closer look at the chemistry of DNA synthesis. So we now have our 3', 5', whatever these numbers mean. And that's the first thing that we want to clarify. For that, we want to concentrate on this ribose. If you are chemically inclined, then you know that this is the closed conformation of the ribose sugar. If there is a closed conformation, then you are of course going to have another open conformation. So the question is now, how do I number these carbon atoms? Here you choose the carbon atom with the highest oxidation state, which then will get the number 1. This is this aldehyde carbon here. And basically the connection is like this between the 1 and the 4, which then gives us the closed conformation of the ribose sugar. And if you are a little bit chemically inclined, then you may have already spotted that this is really not representing the sugar on this side, right? So I have now 1, 2, 3, 4, and 5. And when you look at the position 2, here we have an H here. And here we have an OH at the position carbon 2. So go back a couple slides. Before, when we looked at these sugars for the first time, the naming was 2-deoxyribose for the DNA. And this is where the name comes from. So now we look at the polymerization reaction. And to do that, I'm going to first highlight the carbon atoms that are connecting the 2 phosphates. So here I have my 5' and here my 3' carbon atom. So what happens is simply the free OH at this 3' position will attack the incoming phosphate from this next nucleotide and will kick out the pyrophosphate. So this is basically the cleavage of the high energy bond that then also drives the polymerization itself. Nature has another trick, namely it also uses the pyrophosphatase in order to further cleave this pyrophosphate into 2 separate phosphate groups. And what that does is it removes product from the equilibrium. And according to Le Chatelier, what this will do is the polymerization reaction will be favored in order to, if you will, replace the pyrophosphate that has been taken away before. So this is basically the whole secret to polymerization of the DNA. The important take-home message here is that I have the reaction direction always from 5 to 3', and the important thing is to have this OH group that couples to the next phosphate, and that's it. When we go back to Watson and Crick, what they said at the end of their landmark paper, that the specific pairing immediately suggests a possible copying mechanism as long as this polymerization reaction strictly obeys the base pairing rules. So A to T, C to G. The only thing that you need to do is separate these two and keep the base pairing and fill in the blanks. That's it. And this actually also already gives you the molecular explanation of a mutation. It's simply wrong base pairing. So with this model from Watson and Crick, we can just go back to a cell and understand what it really means passing the hereditary information to the progeny. You simply duplicate your hereditary material. Then you have to find a way how you can separate them between the two daughter cells in order to have exactly the double of the hereditary information you have started out with. You have actually already seen this in action. So this, what is called the mitotic spindle, where the chromosomes are here. So those are the condensed chromosomes. So they are basically just before being pulled apart during the cell division. So this is being basically done by these so-called microtubules. So these are these ropes, these cyan ropes that you see here. So they pull apart the chromosomes to divide up the genetic material. So here I want to emphasize that the whole process, duplicating a chromosome, basically going through a mitosis, is one of the most complex biological processes we can imagine. And in a movie like this, we have to appreciate that we can only see what is labeled and nothing else. So when we go back to our simple schematics, where the duplicated chromosome attaches to these ropes to be pulled apart, and when we just zoom in how they actually look like at the interface, then we are going to find a very complicated picture. So this particular one is from 2019, but you can safely bet that this picture will not look the same in 10 years from now. So much to overly simplified schematics. Speaking of complicated biological processes, so far we have a very elegant concept from Watson and Crick how DNA could replicate. So the chemical reaction we went through, so this is definitely what is not happening inside the cell. Inside the cell, we are going to have to find some machinery that actually does this job. After the landmark paper from Watson and Crick in 1953, the whole world indeed set out to find this machine. The winner of this race was Arthur Kornberg, whose lab published the next landmark papers in 1958, where they have described the purification of the machine and, in the second paper, the requirements of DNA polymerization. And for this accomplishment, Arthur Kornberg was awarded the Nobel Prize in the very next year, 1959. As always, there was a lot of skepticism. When these two papers got reviewed, the referees went that far to say that it is doubtful to even actually speak of enzymatic synthesis of DNA, and calling the enzyme a polymerase. This actually turned out to be pretty spot on. In this case, we are looking at the DNA polymerase 1. But at the end, it turned out that the purified DNA polymerase 1 is not the right DNA polymerase that does the replication. So what happened? For the next 10 years, everybody really thought that the DNA polymerase 1 is the enzyme that replicates the genome in E. coli. About 10 years later, in 1969, scientists discovered a very interesting mutant strain of E. coli with a broken DNA polymerase 1. But bacteria kept growing just fine. How can you grow happily if the DNA replication is reduced by almost 100 times than the normal levels? This could mean two things. Either there is functional redundancy, meaning if one machine is broken, the other one takes over, or this particular DNA polymerase is busy with something else. And the second turned out to be true. If it is not replicating the genome, what is the DNA polymerase 1 doing? You know that you should protect your skin from the sun because the UV irradiation will damage your DNA. So bacteria also have to protect their genome from UV damage because DNA is DNA, right? When this particular strain was exposed to UV, the bacteria all of a sudden died much faster, meaning this DNA polymerase 1 is rather busy with the reparation of the DNA after UV damage and not so much with its overall elongation. Okay, and if the DNA polymerase does not replicate the genome of E. coli, what does? 3 years later, in 1972, Thomas Kornberg, son of Arthur Kornberg, purified the DNA polymerase 2 and 3 from E. coli. Today, we know that there are 5 different DNA polymerases in E. coli. If the single-celled E. coli has 5, how many do we have after a couple of billion years of evolution? Well, so far 16 that we know of. Long story short, polymerase 3 turned out to be the main enzyme that replicates the DNA. As you can see, this particular machinery in the middle is quite elaborate when compared to the others. And why did people miss the number 3? Well, when we look at the number of polymerases in the cell, we see that polymerase 1 is the most abundant one. So that's what you find first. And why that many polymerases? Well, they are involved in different aspects of DNA replication. So here you see the cryptic description of the activities. What this means is that the polymerase are equipped to correct mistakes. Let's turn to bacteria and go step by step to understand what this all means. In contrast to us eukaryotes, E. coli is a prokaryote. One of the most obvious differences between these two is that the prokaryotes do not have a nucleus that separates the genome from the rest of the cell. And compared to us, E. coli has a much smaller genome of about 4.75 million tops. And E. coli is very fast. It duplicates every 20 minutes. That's a very good pace. This means that almost 5 million bases have to be replicated by the DNA polymerase within this time frame. Well, you better have a very efficient machinery for this. Today we know that it is the DNA polymerase 3 that does this job. And we also know that this polymerase is extremely efficient, meaning it can add 1,000 nucleotides per second to the growing DNA. And that's really impressive engineering right here. But there is one other issue that is important. So adding 1,000 nucleotides per second, that's good. But what if Pol 3 interacts with the DNA polymer only for a couple of milliseconds? Asked differently, once the Pol 3 lands on the DNA, how many nucleotides are added per binding event? Basically, what I'm asking is, how processive is this enzyme? Well, it turns out that per binding event, Pol 3 can synthesize on average 85 kilobases long DNA. And this is also very impressive. The question is now, where do we get these numbers from? You can obviously do classical biochemistry in ensemble assays and find out these parameters. And this has been done previously, of course. But if you think about it, especially the question of processivity is not trivial. You have to make sure that the numbers you generate are not falsified from Pol 3 dissociating and rebinding to the DNA. This is where biophysics comes in handy. How cool would it be if you could watch this replication machinery go one at a time? To achieve this goal, you first have to have a good detection method. You are most likely familiar with epifluorescence. You have your glass surface here on the objective, and when you shine the light through, you will excite all your fluorescent molecules and create a background. This is not advantageous, especially if you want to see only a few molecules at a time. The total internal reflection microscopy comes in handy here. So as the name suggests, the physical principle used here is the total reflection of the incoming light. When the incoming light gets total reflected, the electromagnetic wave does not abolish abruptly, but penetrates a few hundred nanometers into the solution. And you guessed it, this cuts down the background to the levels that you can really watch single molecule fluorescence. But for this to work well, you really have to be very close to the surface. So in this particular work, the authors have devised a clever experiment. First, they have attached their molecules of interest to the glass surface for obvious reasons. To have a readout, they have added fluorophores that would intercalate specifically with the elongating double helix. And last, they applied flow for two reasons. One, as we said at the beginning, you don't want to have reassociation of the polymerase once it falls off. When you have the flow and the pol 3 dissociates, it will just carry it out. Second, when you apply flow, you push your growing DNA towards the surface into the turf field. Basically, two birds with one stone. And then you switch on your camera and watch it go. I hope you can see these DNAs that elongate, and if you have done the experiment right, then you can do a very simple analysis. You may have already guessed that these experiments are anything but trivial. If you have done it right, with all necessary controls, etc., you can practically sit in front of your monitor with a ruler and you know your pixel size, you know your cycle time, and make your plot. You don't really argue about taste, but if this isn't cool, then I don't know what is. So far, we have concentrated on the E. coli, a prokaryote, quite simple. It has its DNA in the cytosol, it doesn't have any nucleus, and it has to deal with 5 million base pairs in its genome. But it is very efficient, so it keeps dividing every 20 minutes. But this organism has to master different types of problems when compared to the eukaryotes. The major difference between a eukaryotic cell and a prokaryotic cell is that the eukaryotes have to confine their DNA, their whole genome, into the nucleus. And this, for itself, creates a huge problem for any given cell because it has to deal with this compaction. What do I mean by that? So let's just turn to our own genome and try to understand what the problem is. You may already know that the human genome has 3 billion nucleotides. So in any given cell in our body, this number of nucleotides have to be packaged into the nucleus. Okay, let's try to get a feeling for these huge numbers. I mean, 3 billion letters, that's really a lot. So if you would actually space these nucleotides 1 millimeter apart, then you would span a distance of more than 3,000 kilometers. If I would walk this distance, I would on average encounter a protein coding gene every 130 meters. The gene would span a distance of about 30 meters, but the protein coding sequence itself would be around 1 meter. Meaning I have to somehow paste together the actual protein coding sequence within this long stretch. This is actually what makes the identification of gene function quite a challenge. And it can really take decades from identifying a gene and figuring out its function. I will show you an example for this later. So we're just trying to get a feeling for the length of the DNA in our cells. But even if you do not space these nucleotides apart, as we just did, their end-to-end distance of when you take all the 46 chromosomes in a human cell, it would add to 2 meters. And this you have to pack into the nucleus that has a diameter of 6 micrometers up to 10 micrometers tops. So this would be then again equivalent to stuffing 40 kilometers of an extremely fine thread into a tennis ball. So, and if you would really do this for all the cells in the human body, that would actually make to the moon and back. Now we have a problem. How do we fit the DNA into this tiny, tiny little nucleus? If you're a physics student, you probably already thought about entropy. So this DNA will not lay around like a long, long rod, but there is going to be some entropic effect, right? An entropic compaction. And indeed, if you would treat the DNA as a freely joined chain, then you can actually calculate an entropic compaction that brings down your DNA to 200 micrometers in diameter. This still doesn't fit the DNA into the nucleus, but this helps a lot. So then the question remains, how do I get it down so that I can put everything into the nucleus? Okay, what is the factor that compacts DNA up to the level of mitotic chromosome? So when I say a factor, when I'm looking for something that is functional, then chances are really high that I'm going to end up with a protein. And before I reveal the identity of this magic protein, let me show you what happens when I remove it. The chromosome explodes and the guts are spilled out, if you will. And you can clearly see the DNA threads here in the zoom. So here is the crystal structure of this magic protein that steps in after the entropic compaction of DNA to basically do rest of the work. So the colorful structure in the middle is the so-called octameric histone core. You can indeed take apart the whole complex, and in doing so, you are going to find 4 different histone subunits that form this octameric complex. This basic unit is then capable of squeezing the entropy compacted DNA material into the nucleus. On this slide, you see the different levels of nucleosome-mediated compaction of DNA. The very first level is the so-called beads on a string. The name is actually spot on because if you would take the purified components, the nucleosomes and the DNA, and reconstitute it, you would really see a beads on a string under the electron microscope. This is actually really the beauty. We can take purified components and reconstitute functionality. But at the same time, we also have to be very careful. The next level of compaction, the fiber state, can also be reconstituted, as seen here. The field thinks that this reconstitution may in fact represent an artifact. Nowadays, it is assumed that instead of having an extensive orderly zigzag structure, the fiber stage may be more disordered molten structure in cells. So here we can see that textbook knowledge isn't carved in stone. So why should I care? Why is it that important to know how the DNA is packaged? Well, because the DNA contains all the instructions the regulated access to this information has crucial consequences for the cell. At the end of this lecture, I want to leave you with this. One of the great mysteries in life sciences is how one genome present in the zygote can give rise to such a complex organism with over 200 different cell types. So we figured it out so far that it has to lie somewhere in the genes. And the solution to this fundamental problem is that only a fraction of these genes are expressed in every cell type. So the cell needs a way to regulate which genes are switched on and when. Et voila, some of this regulation indeed happens on the level of genome packaging. The cell provides access or denies access to genes, and an insanely complex machinery is involved in this process. An army of scientists from many different disciplines are working on this topic, including Ulrich Gerland from our physics department. Join me next time when I present a brand new physical concept for DNA packaging. So as always, if you have questions to part 2, please drop me a line.